Okay, if everybody wants to join, we will jump into some questions. And you kind of answered this question already, Dr. Liebke. But Mike is asking what prevents these CAR T cells from killing all the plasma cells? And you kind of just went over that, but I just want to see if anybody wants to weigh in on that in a little more detail.
Yeah, there's unfortunately nothing that prevents the CAR T cells from killing off other plasma cells. So basically, they're agnostic to the type of cell. The CAR T cells will go after every cell that expresses the target, which in this case is BCMA. So any BCMA expressing cell, and most plasma cells, normal plasma cells, express BCMA as well. All of those cells will be destroyed, which is definitely an issue and leads to a phenomenon that's called hypogammaglobulinemia, where the plasma cells, normal plasma cells, produce immunoglobulin. And if you take them all out, then we see that the immunoglobulin levels of our patients are quite low and oftentimes need to be supplemented, at least for a time.
Mm-hmm. Okay, great. And Dr. Voorhees or Dr. Kaufman, any comments on that? On just why isn't everything getting killed from these?
So I get the question, if I'm interpreting right, is why can't we cure every last myeloma cell and cure the disease with CAR T cells? So great question. I think we're all asking the same. There is in theory, there are some BCMA negative plasma cells. And so that could be a portion of it. The other thing is that we possibly haven't used CAR T cell while we've studied CAR T cells in the patients without a lot of therapy. One could argue that we might be using it too late to get every last cell. But I think I actually ultimately don't think with our current technology with the CAR T cells that we're going to kill every last cell. And I think and I don't think we fully understand that. Because there are other diseases like lymphoma and leukemia where we can kill every last cell and people can be cured from CAR T cells. But what's interesting about those diseases is that those diseases can also be cured by conventional therapy. So I just don't think we have the answer for plasma cells yet. One of the things just to remember about plasma cells is that they are hardwired to be long living, because we need them to make antibodies for what we're exposed to when we're a kid. We still need those antibodies to be available to us much later in life. And I think that the hardwiring of the plasma cell was one of the reasons why we have a hard time of killing every last myeloma cell.
Yeah, I think that's a really important point. And it may also have something to do with the fact that the CAR T cells don't survive long enough. I have one patient in particular who participated in a anti-BCMA CAR T cell study and had a response, lost a response, and subsequently is responding to antibody therapy directed against BCMA. So had the cells lasted longer, the target was still there. Had the cells lasted longer, the response would have lasted longer. So I think I totally agree. I think the plasma cells have a very long lifespan, and if the T cells don't match that, then the plasma cells persist.
I think that the holy grail, which we don't have at this point, would be to have a target that's myeloma plasma cell specific, but we don't have that at this point. BCMA is plasma cell specific, benign and malignant. GPRC5D is plasma cell specific. The bispecific antibody cefostamab is a plasma cell specific targeted agent. So we're going to see the hypogammaglobulinemia with those agents, just as we are with the BCMA targeted agents as well. So lots of IVIG to go around for lots of patients.
Interesting. Such an interesting comment. Okay, Marie is asking about availability. So ide-cel was approved by the FDA, so that is an approved therapy. And now I think they're rolling out these different sites. She was asking about conflicts over reimbursement issues. Do you all want to speak to that practically? As you know, these are therapies, as you went through, Dr. Liebke, that it's pretty complicated of a therapy, and you have to have specific expertise at your facility to perform these treatments. So do you want to talk about that and availability? All of you, whoever wants to go first.
Yeah,
Um,
I'll just say that, I mean, I'm sorry, you go ahead.
no, go ahead. No, go ahead. Go ahead.
I'll just say that there is because of all of the, I mean, we're obviously all very excited about it but there's a significant toxicity, and you really need expertise to deliver this. There are programs in place that take a little bit of time and that you can't start putting together until it's approved. And the other issue with the getting it in the hospital is that because it's a very high cost drug, the hospitals and the company have to negotiate contracts for it. So I think that while it was approved, I guess just under a month ago, I think it will start being used commercially, certainly within the month. I know that at our site, we are very close to being able to use the drug commercially.
Okay, and others, do you have comments?
Yeah,
I would agree exactly with what Dr. Kaufman said. It just got FDA approved. You know, the centers need to have this REMS program and training in place in order to administer the commercial product. Even if we had participated in the clinical trials of ide-cel, you know, like we had done here at LCI. So there's some logistical red tape that we have to go through. And I think once we get there and we start, you know, clearing insurance about utilization of ide-cel for our commercial ide-cel for our patients, I think we'll be able to start answering that specific question better. Right now, it remains to be seen exactly what payers are going to do with this information. But it has gotten easier over time for those patients with acute lymphoblastic leukemia and non-Hodgkin lymphoma. So hopefully, you know, that, you know, the barriers that have been broken down in those disease types will, you know, trickle down to myeloma as well.
Okay, great. And Dr. Lika, you had a comment?
Yeah, no, I agree. I think I was also gonna make that comment that you know, luckily there's other products already approved for other diseases, like for acute lymphoblastic leukemia for young adults up to the age of 26, and then for lymphoma. And so at least there's some pathway for the myeloma product already. So hopefully it's not going to take quite as long to be able to give it. And then in terms of the toxicity, I definitely agree that it's important that these drugs are given, you know, at specialized centers with experience, because especially our myeloma patients are generally a bit on the older side. My experience having given CAR T-cell therapy in myeloma and in ALL is that it's actually the myeloma products are better tolerated than the ALL products. But at the same time, I think we have to be, you know, really, really careful and make sure that we're very vigilant as we start using this.
Okay, great. Well, we look forward to it opening up and gaining more experience. I think you've seen that with things like selinexor and things like that. Once you start using it in the clinic with a broader patient population, it's a little easier to know what you have to expect. Okay, Patrice is asking a question. Dr. Kaufman, I'm going to ask you this question. She wants you to discuss venetoclax, dex, and carfilzomib. Like, what type of side effects are people seeing? What's the dosing in that trial? And where is this 3-drug trial taking place? And does it matter if patients have had a prior transplant?
Yeah, so lots of questions there. The trial, I don't know all of the sites of the trial. We have it open. I know it's open at University of Alabama, Birmingham. That Dr. Costa is the lead on this. It uses weekly dexamethasone. There was many dose level study, but the weekly dosing of carfilzomib starting at 70 milligrams per meter, or I think actually it's at 56 milligrams per meter squared. So weekly dosing and not twice weekly dosing. And the venetoclax is 800 milligrams. And I think that study is that now it's a randomized study, and the patients are randomly assigned of either they get carfilzomib and dexamethasone versus carfilzomib, venetoclax, dexamethasone. It's only for 11-14 patients. and it's sort of relatively early in the disease course. They cannot have had or been refractory to Kyprolis previously.
Okay, great. And somebody else was asking about just general side effects of venetoclax.
So the most worrisome side effect from venetoclax came from another disease, the CLL literature, chronic lymphocytic leukemia, and where the cancer cells died so quickly that there was a syndrome called tumor lysis syndrome, where the inside of these cancer cells wreaked havoc in the body, and patients actually died. So in myeloma, we haven't seen really clinical CLS. We see laboratory evidence of TLS, but not people haven't really gotten sick from it. So that's very good news. There's increased risk of infections, lowering of blood counts, and then GI toxicity, some nausea and some diarrhea. My experience with venetoclax is that it's in general very well tolerated if you compare to the whole list of things that we do.
Okay, great. Dr. Voorhees, let me ask you a clarifying question from one of our participants. The benefit of melflufen over melphalan, he's just wanting to clarify. Melphalan's able to penetrate the fatty layer of the myeloma cell. Does that mean it also doesn't penetrate non-myeloma cells?
So melflufen penetrates into all cells well. So the relative differentiation between myeloma cells and other tissues is really dependent on the expression of aminopeptidases inside the cells, and there's a larger number of aminopeptidases in cancer cells, including myeloma cells, than there are in normal cells of the body. So that's the basis for the specific targeting. But obviously there are aminopeptidases in normal cells. There's obviously aminopeptidases in normal bone marrow precursor cells, which is why we see the low blood count issues with this agent. So there's some relative targeting going on here, but it's not the same level of targeting as you're seeing with a BCMA targeted strategy, for example.
Okay. Let me see. Sylvia was asking kind of a basic question about myeloma genetics. How do you know if you have 1114? Someone go over that?
Yeah, so pretty standard test that's done on the original bone marrow or subsequent bone marrows where there's myeloma. And the most common test is something called FISH. In myeloma, we look at the chromosomes. The chromosomes are where the cell keeps all of its genetic data. And we look specifically with this test called FISH for 1114. It really should be in every newly diagnosed myeloma patient. Every newly diagnosed myeloma patient should have FISH testing for a variety of things, including 1114, especially now that we have potentially personalized therapy for 1114 patients.
This is really the only targeted therapy we have right now in multiple myeloma. And another question was there about 414, like what's new on the horizon for 414 patients?
Yeah, there's 414 can have it not always, but sometimes results in the overexpression of a protein called FGFR3. And I believe that there are FGFR3 inhibitors. There was probably 10 years ago, we studied what ended up being ineffective FGFR3 inhibitors, and that wasn't really effective. And so that's where we are with 414 right now. There's a whole group of 414 patients where they don't have upregulation of that protein, where you wouldn't expect inhibition of that protein to be beneficial. But I don't think we're going to find a similar, at least in the same pathway that we saw with venetoclax. I think if we're going to target 414, it's going to be the single protein that's upregulated.
Well, that'll be interesting to see if that new bispecific will have any impact on that then. Going after. Oh, no, that's different. I'm thinking of that different target, the one that starts with an F.
Yes, yeah, our targets are getting much more confusing.
I know.
There's a 5 and there's a G.
I can remember the GPR5CD now, but not the.
There you go. Good job. You're doing great, Jenny.
I think one of the challenges, so for the 414, and there's a number of questions in the chat box or the question and answer box about 414. It's not just FGFR3. There's a gene called MMSET, which affects, the expression of various genes in the myeloma cells, and those are, notoriously very difficult to drug targets. So that's a work in progress. I will say that the 414 translocated myeloma patients are enriched for activating mutations in FGFR3 as well. And one of the baskets in the MyDrug study, that the MMRF is spearheading is actually an inhibitor to FGFR3. So
Yeah.
we'll get some data hopefully from that study.
Yeah, great. Okay, Dr. Lika, maybe you could answer some of these questions and the whole group. So the CAR T cells, can you harvest them early if you're thinking that the immune system has gotten beaten up over time, freeze them and use them later? I know that's a strategy that some people are considering, but you want to weigh in on that. And then for allo CAR T cells, do you have to do any type of matching to prevent any of this potential GVHD idea, kind of like you do with stem cell transplant?
Yeah, those are great questions. So we'd love to be able to collect patients' T cells earlier than what we're doing currently. But because so far, at least, it's only been possible to do this in the context of a clinical trial, and the clinical trials have certain inclusion and exclusion criteria in terms of how many prior treatments, had been required. We were stuck with the requirements of the individual trial. And of course, it's a relatively expensive procedure, so it's likely not to be covered outside of actually receiving the treatment. I personally do think, though, that this would be a major advantage to be able to collect the cells much, much earlier than what we're doing now. And I think it's true for any treatment. Right now, CAR T cell therapy is only approved for patients with 4 or more prior lines, but there are already ongoing trials in earlier lines of therapy, especially for patients who have these high-risk translocations like the 414. And so hopefully we'll be able to move it more towards the front line. But we're currently not able to just say for any newly diagnosed patient who achieves a remission, let's collect the cells now. I think that would be ideal, but it's just not technically feasible, and probably nobody is going to pay for it. I think that's the major issue. Mm-hmm. And then in terms of the allogeneic cells, they are manipulated to reduce the risk of GVHD. So their T cell receptor is knocked down. So there is no special matching that needs to happen, but there's some testing beforehand, before the cells are actually given, to make sure that there aren't certain antibodies, but the cells are also manipulated to reduce those risks.
Okay. Thank you. There's so many questions about CAR T cells. I know that patients are asking, like Annette is asking, how do you know which CAR T cell to give a particular patient? So which one will be most effective? And Jack was kind of asking, do you do ide-cel? Do you do one in a trial? Maybe there's a cost difference because you're in a trial, or is it the technology is going to be changing, as you're. So do you all want to weigh in on that? There are so many questions about when do I do this? Which one do I do? Like, it's so difficult, right? It's a great problem to have, and it's better than not having any options, but it is confusing for patients.
Well, I think that it is very likely that everybody's going to give that there's many answers to that question. So ultimately, there's no way for me to outline an algorithm about which patient should get a CAR T cell now. And so I think we make decisions. I think the first thing you say is a CAR T cell is appropriate. And let's just say we're looking at the labeled indication. CAR T cell is appropriate. We're in as was mentioned, we've had 4 prior lines of therapy. It's appropriate. and it's appropriate from a disease perspective and appropriate from a patient perspective. But the CAR T cells have only been studied in patients who have minimal symptoms and essentially normal organ function. So we take that group of patients, and then we made the decision that it's right for the myeloma based on the number of lines of therapy. And then we make the decision that it's right for the patient based on the patient's performance status and organ function. And then we ask the question, what's available? If we have from my perspective, if I had a clinical trial available, I would recommend the clinical trial, because we know the data on the CAR T cells. The clinical trials are trying to improve upon that. And so I would recommend a clinical trial first. Not every patient wants a clinical trial. If a patient doesn't want a clinical trial, or a clinical trial spot's not available, then we use ide-cel. So that would be my approach. That's how I think about it in answering the question today.
Interesting. Dr. Voorhees, you had a comment?
Completely agree with Dr. Kaufman. I think ide-cel's going to be used extensively for patients who meet the FDA criteria for its use for prior lines of therapy. And there are plenty of those patients that are out there, but ide-cel is the first, across the finish line. And, the data are incredibly exciting. I mean, you look at the data with ide-cel compared to what we saw with selinexor dex, compared to what we saw with melflufen dex, compared to what we saw with belantamab as far as the likelihood of responding, et cetera. So it's an amazing step forward. But there's so much more progress that needs to be made for all the reasons that have already been discussed. So a clinical trial for CAR T is definitely the best way to go when available, because we can do better.
Okay, and Dr. Liedtke, do you want to answer Peter's question? Can CAR T be eventually an alternative to transplant?
Possibly. I mean, I think we're really hoping that we'll develop products that ultimately will be curative. I think, that's really the ultimate goal here. And we know that that's possible in acute lymphoblastic leukemia. And so we just have to improve the product until we get there. So if that becomes, a reality, then it'd be wonderful. I mean, it's we call it a one and done therapy. You give it once, and if it lasts, and if it ultimately cures people, then that's all the treatment that they would need. So that is the goal.
Okay, so interesting. Okay, so Robin was asking if she's a deletion 13 patient but has been on a triple combination using venetoclax with carfilzomib, why is it working for her? You kind of talked a little bit about this at the beginning, that there's a small percentage of patients.
Yeah. So when we looked at patients who don't have 1114, there's a handful of patients who have responded. I also have a handful of patients who've been on venetoclax for a variety of reasons, who didn't have 1114. And we just recently published a paper identifying cell surface markers on myeloma cells that would predict patients responding. And those while the cell surface markers, that group of cell surface markers are enriched, they're a lot of them are in 1114, some of them aren't. And in the group, and what we've learned is that the plasma cells that respond to venetoclax look a little bit more like regular B cells, or like a little bit more like lymphoma cells or CLL cells. And so classically, the 1114 cell looked a lot like a plasma cell and been a little bit like a lymphoma cell. And it's those lymphoid-like plasma cells that are much more likely to respond to venetoclax. And so it's possible that that's the situation we're dealing with. Again, even in the original single agent venetoclax, about 1 in 20 patients responded to venetoclax. And so it absolutely can happen. It's just that when we deal with a drug that has a 1 in 20 chance of of response of benefiting the patient, that's not a drug that we're really seeking, because we have even the lowest amount that we consider moving forward with our drugs is at least 1 in 5 patients. So 1 in 20 patients, we're not really, that's not something we would give to everybody. We want more of a chance of working. But if we could, but there's likely going to be a day where we realize that 1114 is not enough, and we need 1114 plus X, Y, and Z, or not 1114, and we can identify this entire group of people who would benefit from venetoclax. We just don't know them today. And one of the things that's been studied is, remember, venetoclax targets BCL2. One of the things that's been studied by the companies that make venetoclax is patients who have high BCL2. Maybe they respond. We know in our laboratory data that the amount of BCL2 isn't enough to confer sensitivity. But if you take a group of patients who have higher BCL2, that more people in that group will respond than a group of patients who don't have high BCL2.
Yeah. That would make sense. That makes sense. We had a question in the chat also about extramedullary myeloma. Dr. Voorhees, do you want to weigh in on anything about, it's good because I know some of the melphalan results were like particularly helpful for that. Are there other, do you want to weigh in on that? And then are there other options for extramedullary myeloma patients?
Sure. So for the audience, extramedullary multiple myeloma basically just means myeloma that's learned how to grow beyond the confines of the bone marrow. You know, most cancerous plasma cells require on the nutrients within the bone marrow microenvironment to kind of grow and prosper. You take them out, they don't do so well. One of the frustrations back in the day when I actually used to treat myeloma cells in those Petri dishes that I talked about in the lab is that they die very quickly when you take them out of patients. So you have to, work with them very quickly. Extramedullary myeloma has, acquired the right set of mutations to make it independent of those growth factors in the bone marrow microenvironment. These are patients who oftentimes have other high-risk features. They have high-risk cytogenetics, for example. Their LDHs are high. Their disease is growing fairly quickly. These are very difficult patients to treat. And, it is an unmet need, just as high-risk myeloma in general is an unmet need in the myeloma community. So, the fact that we've got a drug combination that does have at least some activity in that group of patients, I think is promising. I will note that in the BCMA CAR T cell studies, in many of the bispecific antibody studies as well, we are seeing activity in extramedullary myeloma as well. So I think we're making progress for those patients, but we certainly have a long ways to go.
Okay. We are running out of time, or have run out of time. So I'm going to ask one last question from Heidi. Can you all of you maybe, compare the risks and benefits between doing a second transplant, like a salvage transplant, versus a CAR T treatment for a relapsed myeloma patient? I think this is a pretty practical and will be a common question.
Well, the side effect profiles of the two, approaches are very different, obviously. So, as far as the second stem cell transplant is concerned, the side effects are going to be very much in line with what they were the first time around. So, significant drop in counts for a brief period of time, significant gastrointestinal side effects from one end of the GI tract to the other end. And, a significant, hit to your stamina and energy level, that will gradually get better over weeks and several months. With CAR T cell therapy, the chemotherapy that it's administered, the fludarabine and the cyclophosphamide, which is most commonly given prior to the CAR T cell infusion, is not nearly as high dose. So it's better tolerated. There's some nausea, some fatigue, but it's not severe. There is some cytopenias, but they're not quite as profound. And the gastrointestinal side effects are minimal. But on the other hand, you've got the cytokine release syndrome that you need to, attend to. You've got the potential for neurologic side effects, that were discussed earlier, so, that there's definitely trade-offs. But, as long as you identify CRS and neurologic side effects promptly and intervene on them promptly, I think CAR T cell therapy can be very well tolerated.
Mm-hmm. Okay, Dr. Kaufman, Dr. Lika, do you have any closing comments about these types of decisions patients are making? It's tough, right? I say see a specialist because, like you said, Dr. Kaufman, like not everybody is the same, and you really need to be seeing a specialist in making these critical decisions.
Yeah, I mean, I think getting that. I mean, that is certainly correct. Getting to that specific question. The other thing I would say, in addition, is that it depends. A lot of it depends on how the first one went, not only from a side effect perspective, but from a length of therapy, length of benefit. So if somebody had an 8-year response, the transplant worked for 8 years, we could expect a second transplant, assuming the myeloma is back in remission, to work for at least half that period of time. Then I would vote for a second transplant. If somebody had a 2-year remission and we weren't, and we were having a hard time getting a remission, then I wouldn't do a second transplant. So I think that so again, can't answer that question globally. It really depends on what's going on in this specific instance, which, as you said, requires expertise.
We say that all the time. Dr. Leakett, I will give you the final word.
Yeah, I think I agree with what's been said. I mean, I think it depends on the toxicity and on the effectiveness of the therapy. And I'll just say that most of our patients who participated in the CAR T cell study had one prior transplant, and most of them said that they thought the CAR T cell was easier than the transplant. But I think that's also that's very subjective.
Mm-hmm. Well, great. Well, we covered a lot of ground today in a very little time. The time goes so fast, and I'm so sorry because we have so many questions that are not answered yet. But this broadcast will be available on our Facebook page, so you can go and refer to it again if you need to, or share it again, and then in a few weeks it will be up on the website as well. We'll also be doing another event on ide-cel with Dr. Nina Shah, which she'll go into more depth. And I just did a recent radio program with Dr. Rajay on that. So we have a lot to learn as patients. Thank you so much, Dr. Lika and Dr. Voorhees and Dr. Kaufman, for joining us today. I think you're spectacular. That's all I can say. And I really appreciate everything you do for myeloma patients every day.
Thank you for having us.
Thank you for inviting us.
And thank you for watching.